Okay, sir. So I guess we are live now.
Should should I come online?
Yes. So we had today registration over 100, and
Great.
it's a good number. It's a good
Thank you.
number. And I think participants are like eagerly waiting for your event today to listen you on
Yes.
on this
I don't
event.
see any participants. Like it's something.
They are not here.
Yeah, okay.
They are on YouTube Live. So we stream our all the events on YouTube Live.
Oh,
So
okay.
yeah, so all the participants they get the link from the live. So I would request our team member Vansi to please once check if everything is okay, and she would give you
Yeah, everything's okay.
perfect.
YouTube Live's perfect.
Okay. So we can start today. So I would start with welcoming all our audience today who are eagerly waiting to hear us with Dr. Sunil today morning, that too on a Sunday. Thank you everybody for joining. And I would also like to thank with the starting itself, Dr. Sunil, to accept our invitation and taking some time out from your busy schedule. Thank you so much, sir.
Thank you very much. First, I comes in a school community. It's really. I hope it will be a wonderful event today.
Definitely. So, without wasting much time, we should quickly now start the event. So, I would like to kindly request Bhakti to please introduce Dr. Sunil today on the event.
So, good morning, everyone. Let me quickly introduce our today's speaker, Dr. Sunil Shetty, an esteemed scholar and seasoned professional in his field. He has done his bachelor's from St. Xavier's College, Mumbai in 2008 in microbiology, zoology, and biochemistry, master's from the Maharaja Sayajirao University of Baroda in 2010 in biotechnology, and he has done his PhD from Indian Institute of Science in microbial genetics in 2016. He has been postdoctoral researcher at Biocentre University of Brazil in 2016 to 2023, and currently he is working as a principal investigator at Tata Memorial Center, ACTREC, that is, if anybody don't know, Advanced Center for Treatment, Research and Education in Cancer. So he has been honored with Indian National Science Academy Young Scientist Award in 2017, M. Srinivasya Medal for Best PhD by ISC Bangalore in 2017, Long-Term Postdoctoral Fellowship PNBO Heidelberg in 2016, and Shama Prasad Mukherjee Fellowship in CSIR in 2010. He has been recognized as Assistant Professor at Uniraga National Institute, Mumbai in 2020. His research interests are regulation of protein synthesis by signaling pathways, alternative translation mechanisms during the stress response, mTOR signaling, and its role in mRNA metabolism. So we are very honored to have you today as a speaker. I'm looking forward for the event. And with this warm welcome, I now request the host to continue the event. Thank you.
Thank you, Bhakti.
Thank you so much, Bhakti, for the introduction. So, to break the ice, I would start it like this: that, sir, do you watch Sunil Shetty's movies? And how do you like them?
Very few, I think. It's. I don't think he's very famous. I guess. Only Hera Feri, I guess. Yeah.
Hmm. Good. So, like, the first question that I would like to ask you today is that what was the first moment in your life which inspired you to pursue career in science? Could be in school or maybe during university.
That's a very difficult question. Like I was thinking about this question that what's the first moment as such. Uh, I can't as such say that okay, this is the first moment.
Mm-hmm.
Only thing is in childhood when the school time, like I was very good in drawing.
Mm-hmm.
Okay, then my science teacher used to say that oh you are very good in drawing, so like you can be a good like MBBS or some student where they have to do a lot of sketches and stuff. So that can be good for you like
Mm-hmm.
that. So that's a I think a little bit idea in my mind came that okay, maybe in MBBS and or something because of that school teacher when I was in the sixth grade.
Mm-hmm.
But then apart from that, but then till 12th, I was interested in pursuing MBBS.
Mm-hmm. Mm-hmm.
So as such, research was not ever like a plan in my mind. Okay. But then I think MBBS, I didn't manage the government seats. So like then I said, okay, then I don't go for private quota. So then I decided to pursue BSc, and that too, I must say that I had a poor economic background, so I
Mm-hmm.
always had this in mind that job security as a point. Okay, so that's why I chose a microbiology because somebody said even in case you don't want to study further, you can find a job after BSc into microbiology. I said okay, that sounds better. So like that. But then as I dwell into like bachelor's, masters, I think masters is the time where I work with the biofilms in bacteria. That time I started enjoying really that like whatever the failure and all. It didn't daunted me. It actually made me like a challenge, like take the challenge further, like that. So that's the moment I would say, like the masters, the dissertation time, where I thought, okay, research will be fun for me. Then I thought of doing the PhD because of that. I would say.
I see. So so during masters biofilm, so definitely MSU in microbiology department and biotech, most of the PIs work with biofilms. So
Biofilms, yeah. Like that time in biotechnology, only Professor Shah was working. Others were doing
mm-hmm.
immunology and some lipid metabolism and all, prions and all. Very exciting that time, actually.
So like uh, exactly at what time did you decided okay I want to do a PhD, like before masters or like
No,
during
during
PhD?
masters.
Uh-huh.
Till masters also, I had a because that's why I pursued biotechnology,
Mm-hmm.
thinking that I can jump into industry as a
Mm-hmm.
backup.
Mm-hmm. Mm-hmm.
But during the masters, I think my like during this dissertation and all,
Mm-hmm.
my interest to do PhD. There is. Before that, I never thought that I will do PhD or something. Like
Mm-hmm.
first of all, even during BSc, I was not so much aware about PhD programs and all. So
Mm-hmm.
it's I would say masters which determined.
Master's degree
Like
and
a late comer, I would say.
I see. And can you describe that what hurdles did you face during your PhD, like, and how did you overcome it? Maybe one or two.
So when you say hurdle, is it about the like the project or like getting
Yeah.
into the field project?
Yeah,
Okay.
project, and yeah, also how did you manage your personal life
So
during
the thing is, there are two aspects. Like with respect to overall organization, I would say that I was a bit of haphazard personality because I get too many interests. Okay, so like I used to do multiple projects at a time. So then focusing on one thing and getting something done was a bit difficult for me. So then my boss used to sometimes tell me that Shetty, don't be over ambitious. Try to focus on something. That was one aspect. Okay. But in PhD, the first project which I took was about the identifying a suppressor mutation in a bacteria. Okay, it's like you look for another compensating mutation in the bacteria genetics. And that time we used to do mainly classical genetics, like you do the transduction, mapping kind of technology. So there, I was struggling to identify because I knew that okay, this mutation lies in this area, this locus. But then sequencing and identifying that was a challenge because that same gene was existing in 7 copies, multiple copies. Okay, so that's why to now among the 7 copies, only in one of them has the mutation, and to identify that was a tricky because even NGS with the shotgun and all you can't make out because it will not map. Okay, this is the gene which has right. So then, but we are sure with our genetics that okay, this is the location where you should have a mutation. So that belief was there, but we were not able to identify. Then, like it almost took 1 year more than one. Like it's almost at the end of the second year, I could map it just because I could. What I tried was like I was keep trying multiple approaches, but the best thing the thing which worked was that among the 7 copies, I looked for one nucleotide which is variable. So then I designed this gene-specific nucleotide, like a primer, which will bind to that 3' end, because you know that 3' end of the primer is very essential to do the amplification. So that primer design to focusing on that gene with that 3', worked. Then we could identify the mutation. So that was like okay, because after that, the story development was like once you know the mutation, we could go through the mechanism within 1.5 years, and we could publish that paper. But that was like a roadblock for me.
Mm-hmm.
That I could still remember that coming up multiple different approaches and all was very fun. That time maybe frustrating, but yes, was a fun thing.
But now, when you are a PI, how do you look that hurdle?
So that's what. As a PA, now I smile at it. Okay, definitely. But it was a challenge. The only thing I would say that I never got stuck with one approach.
Hmm.
That's the one thing I also suggest other students also, that never stick to only one experiment. That's the only way to get that answer. Try multiple approaches. That's important. You have to be innovative little bit and try different. Maybe sometimes it's a bullshit idea, but still try. So that I would say, don't just rely on plan E. That's the thing.
I see. That's really good, and I think it's very common even in PhD to get the results during your last 2 years.
Correct. That's true. That's true. Yeah.
Mm. So, sir, now since you were
Shetty. Yeah.
since you are now since you in your journey when you were finishing your PhD, like, did you decide to pursue a postdoc, like, or
Yeah, during once during from the beginning, like the PhD journey only, I had this mindset of going for the postdoc. Okay. That's mainly because my personality, I would say, that I don't see myself following something very nice. Like if you think in the industrial setting, I will be a very bad member for industrial. Because I'm really not very well organized. So my mind keeps on pondering around, and like the new ideas excites me. So that's why I felt that I'm not a person who can dedicatedly work for somebody. So I think this is
Within.
academic field is one which I might survive. Then I said, okay, that's good for me.
I see. I see. And how was the cultural experience? So can you share like the journey when you came here, and then what did you experience, and how did you cope up with I mean, considering everything during your postdoc?
So the journey from PhD to postdoc,
Yeah.
the postdoc, yeah, like definitely cultural shock was there, because I went to German speaking part of Switzerland. They are little bit more like a methodological, like very. I don't
Organized.
say that. Yeah, very ordered. Like for me, it's like complete opposite of me. Like I'm not a person who can plan my day before. Okay, so and they believe in planning and properly. I was not that kind of personality. So that was a culture shock. For them also it's a shock to see me doing my things, managing things without any plans. So that was the thing. But as such, language was a barrier, I would say, there in like settling initially. But it took some 3-4 months for them to adapt to me, or me getting into them. Like so, that was the time. I think they also needed some time to adjust with me. But yes, but then it was really nice. Okay, so it's just that me and my boss mentor there, Michael Hall. So we had a different ideology about the science. So we always had arguments, but otherwise it's fine. Like he wanted more like hypothesis-driven science, and I was more like a data-driven science. I was like always, I need data to make hypothesis. Blah, blah. So we had these arguments, but and then because I had a MBA fellowship and all, and since I had written my project, so I had a freedom to explore my idea. So that was the thing. But um, my boss was very good in killing all the ideas. So something which survived, we could manage to publish. but was nice. I think I learned a lot
Mm-hmm.
there as a postdoc.
Mm-hmm.
Yeah, very helpful for me.
Mm-hmm.
Definitely.
Mm-hmm. I see.
Also, I would say that what I learned in Europe is giving some value to ourselves.
Yeah.
That's the one thing I would must say that changed my perspective, because in PhD and all, I was more about the workaholic, I would say. Okay, more, but then in Europe that mindset little bit reduced. So I felt that okay, we should also give some value to the life,
Mm-hmm.
our own life. So that was a good advantage, I would say, doing the postdoc in Europe.
Mm-hmm. So internationally known work-life balance of Europe,
Yes, yes,
and
that
it
yeah,
helped you, and it helped you
that helped
positively.
that helped me positively. So I tried to bring it here also
Mm-hmm.
as a PI. Let's
Mm-hmm.
see how long I retain. That I don't know.
I think you will do really good. I'm sure.
We'll try. Yeah.
So after finishing your postdoc, like what motivated you to return back to India and become a PI?
So like I was like when I went to Europe, I had this mindset that I will be coming back.
Okay.
Okay,
Mm-hmm. Mm-hmm.
that was the mindset I had initially,
Mm-hmm.
and that all my friends also were like even Michael all was wondered like why are you so sure that you will go back and all. But
Mm-hmm.
I had this feeling, but only after COVID duration, I had little bit of like I traveled a lot in Europe and all.
Mm-hmm.
That time I started liking Europe as well as a like a because there is you have more peace of mind. So that I felt that in Europe I can stay. So I thought of little bit. There was a consideration started to stay even in Europe,
Mm-hmm.
but then majority my interest was in India. Coming back to India
Mm-hmm. Mm-hmm.
first, the main reason I would say that I think I belong here more than any other country. So that one belonging part is the major reason I would say,
Mm-hmm.
because wherever I am in that country, I had so many friends and everything. Still, I think that I have to put extra effort to be there. And
Mm-hmm.
here, I don't have to. I just have to be myself, and it's fine. Like no one will judge me.
Yeah.
That like that part somewhere. That part is that okay. You are here now. You are at home. Like that
Mm-hmm.
feeling comes here. So I think that's one thing. Second is, as I said, I am from a little bit economically poor background, so I
Mm-hmm.
have obtained whatever my journey is
Mm-hmm.
support from here itself.
Mm-hmm.
So I had to give back also to the society. I thought whatever I do, let's do it here. Okay, so there are a lot of issues are here, but definitely, but I think it's fine. That's our own problem, so we have to manage that. So that was the second feeling I had, and that is family because family one of the like my wife wanted me to
Mm-hmm.
come back to Mumbai, so
Mm-hmm.
and as she wished we found a position here only. So that's another factor. Okay, Mumbai, then okay, let's family is close by. And the fourth one is when I tried in Europe, as I said, right? Like I had a like things were going on with the Stockholm University and all, but
Mm-hmm.
the job security was
Mm-hmm.
another factor determined because in Europe most of the programs as a PI
Mm-hmm.
they are never permanent. Like after 5 years, 6 years, you have to move, find another one. So
Yeah.
you're never settled.
Yeah.
Like maybe good for keeping yourself pushed,
Mm-hmm.
but sometimes some people also like me want to be a bit settled also. Like I don't want to keep running around. So that's why here I had a permanent position offer like career track. So I said okay, that's good for me. So then I think those are the four key factors I would say
Mm-hmm.
determine my choice.
Mm-hmm. Mm-hmm. And so, like, were you open to even explore in other places as well, like apart
Yes,
from Mumbai?
I was like my choice was yeah you can like my choice was South Indian part mainly
Mm-hmm. Mm-hmm.
like the Bangalore and Hyderabad. I would have like, but I didn't apply. Like I
Mm-hmm.
applied IIC, but otherwise I didn't explore much because this offer was very sudden. Like I was not ready while my postdoc, like my manuscript was under review,
Mm-hmm.
and then this position I got it,
Mm-hmm.
and they said like within 3 months you have to decide whether you want to join or not. So
Okay.
then very tough decision to make. Like it was really
Leaving everything and coming back.
yeah. So yeah, then but then I decided okay. For overall, if I say It
Mm-hmm.
was a positive, like a plus direction, I would say,
Yeah.
positive decision. Yeah.
Yeah. Yeah. Yeah. I think so. Like, how were your initial days when you started your own independent group? Like now you are the PI, and
Yeah.
especially the challenges that you faced, like day to day life, and what are some memorable moments that you would always cherish?
Okay. See, the first of all, jumping from Switzerland in mid-February, from Switzerland to Mumbai. It's like from cold shock to heat shock directly. Okay, and this campus was really like a dried place during that time.
Mm,
So summer
mm,
already started here. So when I jumped here, starting was a really shock for me, and I hadn't visited Attic before. Okay,
mhm,
so I didn't have like during my bachelor's once we had come for their annual day or something. Others
mm,
I didn't have much idea about Attic, the campus.
mm,
So that was thing, and then the lab was not ready. Like I had to renovate the lab and everything.
mm,
Like it took
mm,
settling that lab. I it was thanks to my trainees initially who actually helped me a lot in settling the things, managing things and all. So hurdle was there in setting up the lab itself,
mhm,
getting the equipments and all. And here admin aspects are very slow.
mm,
Like I knew little bit of Marathi,
mhm,
so it helped a lot me to communicate with people and get things done a bit faster. But yeah, it took minimum 6 months, I would say. Like in September, we started as such,
mhm,
like from April to settle the lab
mhm,
properly.
mhm,
But then it was going nicely further. Then it all student driven now. So
mm.
I don't say that I really contribute too much, but yeah, I think I do my part little bit. But I think students are good, so they take care of the lab mostly. I don't go with the organization and everything, so they are the bosses in the lab. So they take care. So
That is really good. I think that's a sign of a good
yeah,
PI.
so they help a lot. So I think it's going well so far. I can't complain so much.
I see. I see. So now, since you are a PI, you also have students. So, like, you also see your own PhD journey, your own postdoc journey, right? So, like, out of your maybe I would say 10 years, yeah,
12 years, I think. Huh?
yeah, more than 10 years, yeah. Like in hardcore research, like, what advice would you like to give to students like us?
Okay, so advice is a big term. So the thing is, I keep telling my students also that
Mm-hmm.
first of all, never stick to one project.
Mm,
For me, always keeping a backup plan is always important. So I
mm,
try to push my students also to do multiple projects at least in the beginning. At least when you are in the start of your PhD,
mm,
try multiple projects and find what's your interesting question. You have to be actually really motivated to really struggle for that question. You know
mm,
that if your boss says okay you're doing it, that may not work so much nicely. So try to find the question which whatever it's in the lab, try to
mhm,
do whatever something.
mhm,
Don't get committed in the first day one that oh I will start working on this project. So that's not a good idea. I would say that take your time, decide okay this is really the thing which you want to pursue. You will be satisfied after your PhD. I think that's important. Like spend 1 year, it's fine,
mhm.
but at least within 1 year you figure out what you want to do and then jump into it. But then maybe you have to focus on it, otherwise you will be like me. Like I got the lecture after 2 years, so that's it's better to take some time and well into it proper. Yeah, that would I would suggest.
Mhm, mhm,
And
mhm,
second thing also I would suggest that own your project. That's important. Like don't work for your boss. That's important. You have to own your project. Okay, that factor is important because I still remember that during my PhD, if my boss is to come up with the idea for my project, I felt bad. Why didn't I come up with that idea? That was my like the competition. I was competing with my boss itself. That I should bring that idea should have come to me first than my boss. So that kind of belongingness to that project is needed. If you can't do that, then I think you will not enjoy your journey because you should, if you're really into that project, definitely, even you in dreams, you sometimes get the experiments running around and all like, oh, morning, okay, I have to go for this kind of that right now I remember those days. Like, okay, I could not sleep because of the experiments that were going on in my brain. Okay, this is I should try this one. I should try. So those kind of environment should come as a researcher. If
Hmm.
you just go to lab and 9 to 6 you work and come back, then I think you should think about your profession carefully. That I
Hmm.
would say.
Yeah.
Yeah.
I think I heard from somebody. I was listening to some podcast or something, and they said in that uh, you know PhD is like for yourself. You do PhD for yourself, not for anybody else.
Anybody else?
And
Yes.
whenever you feel like working, don't see time. Like don't see it's 2 o'clock or 3 o'clock. And if you
Mhm.
don't want to work, then don't work at all.
At all. Yeah, that's correct. True. And that's what sometimes nowadays PhDs are, I would say that many people are like, it's a matter of status kind of thing. I think many
Yeah.
students think, oh, getting a PhD is the big thing.
Hmm.
It's not at all. The degree doesn't have any, like, once you have the open defense and you say, okay, you are a doctor now, you feel nothing. There is no special thing about being doctor.
Mhm.
It's all about your journey, how you struggle, how you manage things. So that's actually the thing, and you should enjoy that part. I would say that's the only memory. Nobody cares when did you get that degree. Like I don't even remember my open defense.
Mhm.
That happened on Sunday or something. Like like only 3 people were there and I got my degree. So that's it. Nobody cares so much. So
I see.
the journey should be memorable. I would say.
I see. I see. So, sir, like since you were talking also about work-life balance,
Mhm.
so like how do you currently meet the demands of the lab work and also personal growth, also,
Okay.
and at the same time mentoring students as well? How do you manage this all? And do you have like a hobby or something like that?
That's a tricky question for me. I should say that hobby as such, I don't see any hobbies in my hobby is to actually interact with the people. Okay,
Mm-hmm.
I like to interact with people, so I
Mm-hmm.
interact with my lab students.
Mm-hmm.
Like I don't know whether it's a mentoring, whatever. I spend time with my students is what I believe.
Mm-hmm.
But also I interact with other students. Like I'm open to discuss with other students also. So
Mm-hmm.
that part is what I enjoy more. Also, little bit organizing events and all. I try to push in the
Mm-hmm.
student community. That's the
Mm-hmm.
thing. But I must say that I don't spend too much time in the lab work.
Mm-hmm.
So I give my students take care of things. So I don't go there and do so much. If they need something, they drag me to the lab. So then I tell them that okay, make me work there. So that's
Mm-hmm.
what they have to make me work. Otherwise, I'm not a person who will go to lab and they will generate data. So I can't do that because I trust my students' data more than myself. So I think
I
that's what.
see.
So I get some time since I don't do lab work so much. I think I have some time.
I see. I see. So, have there been any mentors in your journey? Like maybe one or two mentors which you would have felt like, yeah, like they really helped you a lot. Or maybe being a PI, do you have a mentor?
That's what I like my PhD mentor. I would say was
Mm-hmm.
the real I would say motto, and he gave me a very positive vibe about the research. The mentor was good, and he gave the lab environment so nice, like vibrant. We had a freedom to ask questions, and he was very open to the arguments. Like he never felt it. Oh, this student is being rude or something. Nothing. Like we were always able to argue with our ideas. So that positive culture actually helped me a lot. I think that's why my I would say the Umesh Wasne in ISC was a real mentor for me, at least with respect to managing my lab and all.
Mm-hmm.
But then also I should give some credit to my professor Michael Hall, where I did my postdoc.
Mm-hmm. Mm-hmm.
His ability was to look for the the good question,
Mm-hmm.
because where as a researchers we can get thousands of questions.
Mm-hmm.
Like if I do any, you start with any field, you will get questions, not
Yeah.
that. So you can do keep on, but then he is like not every observation you have to explore
Mm-hmm.
every idea. So
Mm-hmm.
he always tried to ask for a bigger question, bit
mm,
more challenging and bigger questions. So I think he pushed a bit
mm,
than me getting into, like, because I was, as I said, during PhD also I had multiple ideas, try
mm,
to explain the bacteria.
mm,
Like I could continue any multiple direction, but then he actually nailed me down. He said that, Shetty, for other students I had to motivate, but for you I had to cut you down. So I had to keep you focused on something. So that part he did well. So I think that's also important to know where you should focus your energy. I
mm,
think both mentors help, but overall right now, whatever the lab environment, everything, I think Umesh was had a big impact. I still, if whenever the issue something comes, even if it managing or anything, I still contact them. So it's I still have them as a mentor for me, I would say.
I think that's one of the really good thing. And most of the students, I mean, I would say, including me, all the PhD students, they usually struggle in this scenario. They get three observations and they're like, these three all look interesting, but okay, which one works
Which one to
much more ahead is we don't have that vision.
correct? That decision is very critical, and it's very difficult also to choose. Like
Mm-hmm.
now I work with the multiomics, like this proteomics and aspect. You
Mm-hmm.
get hundreds of targets
Yeah.
with on which you have to focus is a big question, always troubles. Then yeah, you need something more work to focus on
Mm-hmm.
something. Yeah,
Mm-hmm.
that is
Mm-hmm.
it.
I see. I see. So since you talked about like uh, proteomics and all, so I would like to ask you that what motivates you to explore the non-canonical mechanisms in the protein synthesis, the research question. What is interesting
Yeah.
for you?
So the non-canonical, again, this concept came to my mind during my PhD itself,
Mm-hmm.
where in the Washington's lab, we were working with the bacterial translation initiation. Right? So that was the main focus of the lab, how the ribosomes recognize the start codon. But then there we had observations where you can also, under different stress condition, you
Mm-hmm.
don't recognize its original start codon, but it
Mm-hmm.
can recognize something else, some
Mm-hmm.
other from there. You can same mRNA, but you can produce a different protein, just changing its start codon recognition. Right?
Mm-hmm.
So that was the interesting concept, and there are multiple papers on it that under stress conditions there can be this non-canonical translation
Mm-hmm.
producing new proteins and helping the cells survive. So
Yeah.
that concept had in my mind from my PhD. So little part that concept remained during postdoc also. So then in postdoc I started exploring yeast and cancer cells. So there that concept was like we had some support for that concept. So that's why now in my own lab I'm trying to explore that part more detail.
I see, I see, I see. So, yeah, most of the cancer works are done on yeast.
Yeah.
Even in our institute, we work on cancer. So they grow like tons and tons of yeast in big shakers.
Yeah, but for me, I use yeast because I feel it's more reliable system
Yeah,
than building a story on a cell line. I
yep.
somehow I have this feeling, that confidence, something which is very consistent from the
Mm-hmm,
evolution mechanism.
mm-hmm.
That is will be worth, but it doesn't mean that only that we are exploring. We also explore cancer specific aspects. For
Yeah,
that, we use the animal models and all.
yeah,
But yeast is, I think, is a good system for exploring something new. I would say.
yeah. Because it's also very less complex and also it's a smaller genome as compared
Yeah,
to the mammalian system. So absolutely, yeah.
correct.
So like you made some PPT as well for
Yes,
the
I
event.
you told me to prepare 10 slides.
Yeah,
Okay.
that should be enough for students to see your work as well. So it would be great if you can present us with the small presentation and overview of
Okay,
it.
I'll try to share my entire screen. Right?
Yeah, sure.
Do you see presentation now? Dhairya?
Yes, sir. Yes, your presentation, I can see it.
Wait, is it in a presentation mode now?
Yes.
Okay, so I
Looks
can start,
really
right?
good.
So,
Basel Bio looks really good. Basel Biocentrum. I visited Basel just last week for the
really, oh,
Christmas
nice.
market. For the Christmas market.
Oh, acha, yeah, Christmas market started there, right? Yes. Yeah.
Yeah.
So can I start now?
Yeah. Sure.
Okay. So hello all. I think this is the topic which I would like to talk is about the non-canonical protein synthesis mechanisms and the stress and disease conditions. So this is a brief journey about my journey that I am from Udupi, a small town in Karnataka, and then I did my bachelor's from Mumbai, and then a master's in Baroda city. Then I did PhD in Bangalore. So and then I went to Basel and again back to Mumbai. So I have this favorite dialogue saying that it used to be Bombay, Baroda. So Bombay, Baroda, Bangalore, Basel. So B series. I always travel in B series. So said that now protein synthesis. My exposure to protein synthesis started from the PhD, I would say. And you all know that the material, the information in DNA has to be converted to protein for most of its function, and that is mediated via mRNA. So after transcription, we get this mRNAs. I think most of the students have studied the transcription translation process, and in textbook knowledge that mRNA has a start codon and a stop codon. So between these, the triplet codons are read by the ribosome with the help of tRNAs, which recognize the start codon and the other codons and bring the amino acids to make a polypeptide. So this is a very well known concept, I think, for every students. So here, but if you dig into more, so this process is conserved from bacteria, right? So all the from bacteria to mammalian cells, it's conserved. And my PhD journey started with the bacteria, and where we were like the translation, you can divide into four large steps: that initiation step, elongation, termination, and the ribosome recycling. And initiation is the one which is very crucial because that determines where your protein synthesis begins. So that's why it's called as a rate limiting step as well. So once you recognize this, then you keep on synthesizing the protein. So the same mechanism is also conserved in eukaryotes as well. The most of the steps like elongation, termination are pretty conserved, but the initiation, if you see, it has a little bit variation. And this initiation in mammalian cells, you would say that, or eukaryotes in general, has a scanning mode of initiation where the initiation starts at the 5' cap structure where the 40S is loaded, and then they do scanning. This is the canonical translation. So, and there is evolution of this, like there are a lot of factors involved in this process, and also it's regulated by various signaling pathways. So, now if I say that the canonical translation, this is what, as in the textbook context, that you have this cap structure, from there the start codon recognition happens, AUG, and then the whole open reading frame is translated, and then the stop codon, at the stop codon, the ribosomes are released. That's the canonical for us. But when I say non-canonical, that means apart from this, there are multiple other ways in which the same message can be read in other language. So that's these are the certain examples, because the same message, if you have a internal ribosome entry site, then it can lead to a shorter peptide from the another AUG internally. So you can get a another protein. Even it doesn't have to be in film, so you can get another protein. There are mechanisms for ribosome shunt where the mRNA, some part is skipped, and then you start with the another region. That's called a ribosome shunt. And there's leaky scanning where, although you had the main open reading frame, but the internal start codon might be recognized more, and you have another reading frame which is recognized. So this is called a leaky scanning, and there is a non-AUG initiation which we work during my PhD also. That instead of AUG, ribosomes might recognize some other codons like CUG or something, which is not in the same frame, and completely from the same mRNA you can synthesize a newer peptide. Similarly, you have other options like reinitiation kind of thing, with the two proteins might be made from the same mRNA, or the frame shifting, where after certain translation, the ribosomes might change their frame because of which now you have a different peptide synthesized. And these are the like such frame shifting, with the coronaviruses and all, that there is like some certain virus undergo a programmed frame shifting just to synthesize the newer proteins. Apart from that, stop codon read through is another mechanism where instead of stopping at the stop codon, it continue with other codons like downstream. It keep on continuing the synthesis, because of which you can produce a longer proteins which will have a different functions. That's the stop codon read through. Or stop carry on, where you stop for peptide, but immediately you start again another peptide synthesis. That's the stop carry on. So such multiple mechanisms are possible. This all we consider as a non-canonical mechanisms compared to the previously textbook knowledge. So the beauty of this mechanism is that you don't have to make a newer mRNA. With the same mRNA, you might produce a new other type of proteins. And our hypothesis that under stress conditions and all, and even the disease condition, this kind of non-canonical translation might play a role in contributing to the disease progression. So, if you come to the role of protein synthesis in cancer, the earlier notion is that the cancer cells have a higher protein synthesis, because it's known that the ribosome, like the ribosome synthesis, is very high in cancer cells, and also the nucleolar structures are aberrant. So also the pathways which are interested in which lead to the upregulation of the protein synthesis also hyperactive in most of the cancer. So this general notion is that cancer cells, since they proliferate a lot, they also have lots of ribosomes and they do lots of protein synthesis. That was the notion. But now there are counter notion that it's not necessary that you have to have more ribosomes and more protein synthesis for cancer cells to survive. So we are in that, our lab observations go in that direction. So as I said, the main notion comes because many of these pathways, which are oncogenic pathways, are also known to promote the translation initiation. Okay, and based on these, there are multiple drugs now under trial which are inhibiting the upstream signaling pathways which promote translation, or even the inhibiting the canonical translation, where you can block the initiation or something, is all going on. So this is what the current trials are going on. But the major hurdle here is that if you directly try to block initiation or something, there is still a possibility that such non-canonical mechanisms can compensate for that. So that's what we are interested, because the mTOR inhibition and all still have not given a very high success in many of the cancers. So we are trying to explore whether such compensatory mechanisms or the non-canonical initiation or the translation helps cancer cell to survive. So that is the question which we are exploring in the lab right now. So among the oncogenic pathways, our lab is mainly interested in the pathway called target of rapamycin signaling. So it's also called mTOR in mammalian cells. So the rapamycin was a drug which was very long back discovered in Easter Island, and it was initially used as an immunosuppressant. Okay, and it was during the kidney transplantation and all they were used, but then they had their own toxicity. Right now, they are not the preferred one. Some other analogs are there. But rapamycin actually, they didn't know that what it actually has the target. So then my postdoc mentor, the Michael Hall, in his postdoc actually started exploring the mechanism of the rapamycin, how it inhibits cell growth and all, by using yeast as a model system. And in yeast, using the genetics, he could identify a gene called target of rapamycin, two genes, which gets inhibited by rapamycin with the help of another protein called FKBP12. So this complex FKBP12 rapamycin blocks this kinase. And then much more further studies have shown that this TOR kinase exists as a two complexes. It forms two complexes, TOR1 or mTOR1 and TOR2 or the mTOR2 in mammalian cells. And mTOR1 is considered as a master regulator because it senses various conditions of the cell, like if there is a shift in the temperature or the nutrients are available or not, growth factors are available or not, and so the pH and all. So It's a sensor for the multiple stimuli of the cell, and they accordingly the activity is regulated. And the top two is mainly activated by the growth factors, as well as it senses certain stress conditions such as osmotic stress and all. So the both the pathways play a major role in determining the cell growth and metabolism. And mainly the top one is well known to promote translation as well as ribosome biogenesis. And also it's famous to inhibit autophagy. Many people explore this aspect as well. mTOR2 is the one which is bit less studied, explored, I would say. But here they are already known to affect the actin polymerization and the lipid synthesis and chain cytosis processes. So this is the known aspect. So now, with respect to the protein synthesis, how mTOR1 promotes translation. This is the like the simplistic form of the mTOR signaling, I would say. It's if you go in depth, it's a very complicated pathway. So, but for the cartonic purpose, both mTOR1 and mTOR2 are under the PA3K signaling, which is a lipid signaling. And upon growth factor stimulation, PA3, which activates the AKT kinase, and by inhibiting a repressor protein called TSC, it activates mTOR1. Both nutrients and growth factors are necessary for the activation of this kinase complex. So there are two important repressors of this pathway. One is P10 and the TSC. Both negatively regulate this mTOR1 signal. So now, upon nutrients and growth factor availability, what does mTOR1 do is, first, it phosphorates directly a protein called 4EBP. It's a repressor protein which usually prevents formation of the initiation complex called EF4F. So this inhibits the EF4F complex. So when mTOR1 phosphorates it, it is kept away so the initiation complex can form better and then the fortress ribosomes can be recruited. Apart from this, mTOR1 also activates a kinase called S6K, and this kinase directly can phosphate certain initiation factors and promote translation initiation. Apart from that, it also can promote elongation, translation elongation by inhibiting a kinase, which is a negative regulator of the elongation factor. So via this mechanism, both initiation and elongations are promoted by mTOR1 complex when you have a nutrients and growth factors. So now, if this is the case, how do you see this monitor the effect of this torque one on translation? For that, it's a classical method which we also use in our lab, is doing the polysome profiling, where you can take the cells which are the growth condition and then trap the ribosomes on their mRNA using a drug called cyclophosphamide in eukaryotes, and then get the cell lysate from these cells and then run those samples on a sucrose density gradient, where with this density gradient you can separate the different populations of the ribosomes, and then you can monitor there using the UV absorption. So typically you get such a profile indicating the active translation, where you have a monosome ATS and then the polysomes which indicate the active translation. But if you now inhibit TORC1 using rapamycin suppose, what you see is that reduction in these polysomes to a minimal amount and then increase in the ATS population. So this is the response you get when you inhibit TORC1, and it's an indication that translation indeed inhibited by upon inhibition of the TORC1 complex. So this is the tool which we use mainly for monitoring the translation. So now, since as I said, mTOR is known to promote translation, and mTOR is also known to be upregulated like 40 to 50% of HCC patients. So we asked the question, how does it actually lead to the cancer? So that was the question which are interested. So here, what we have done in this was slide generated in my postdoc lab, Professor Michael Hall's lab, where they deleted these two repressors, TSC and P10, which are known to inhibit the mTOR1 and mTOR2 in all the tissues. So here, what they have done is to delete these two repressors only in the liver. So if you delete these repressors, especially using the albumin 3 and double knockout of this will develop the HCC tumors within 20 weeks. So it's like around 5 to 6 months. You see that the tumors are developed, and you can now analyze their proteomics or the protein status, everything using this as a tool. So this was the nice tool we had. So then we ask question, what happens in these tumors? How is the translation? Because as per the mTOR known knowledge, you're supposed to get the increased translation in these tumors. So we did isolate, like compare the controls liver and the tumors from this mouse model, and then analyze the polysome profile. Surprisingly, what we see is not the increase, but these red lines are for the cancer, the polysomes uptake from the cancer cells, and the blue lines from the control liver. What we see in both fed and the starved conditions is that polysomes are reduced in the tumors obtained from this mouse model. So it's a counter notion that you have the mTOR hyperactivation, which is known to promote translation, but in these cancers, we see that the ribosomes are less, or even the ribosomes are less, as well as translation is less. So then the question is, what does it mean? So further, we had confirmed that whether indeed it is true. For that, what we do is another assay called the puromycin incorporation assay to monitor the name translation ongoing. So for that, because puromycin is a drug which can occupy the ribosomal A site and tag itself to the ongoing peptide. So during the translocation event, the peptide can be get tagged to the puromycin, and those peptides can be released now. So this will not tell about the stable already synthesized protein, but ongoing protein synthesis you will get an idea. So we did that experiment that again the tumors and all. In situ, we tried to do the puromycin incorporation and ask the question whether is it really translation going down in the tumors. Yes, that is the case. That compared to control, we see a dramatic decrease in the puromycin incorporation in these tumor cells. So that's again a supportive for a notion that in these cancer cells, the translation is less, although they have a hyperactivation of the mTOR signaling. But what is surprising for us was the phosphorylation of EF2 alpha, which is the canonical initiation, like the initial factor, which brings the tRNA to the ribosomal P site. That initial factor has this phosphorylation at serine 51 high increase, and this increase in phosphorylation is actually known that it inhibits the activity of EF2 alpha. So that means indirectly there is some signaling which has now led to the EF2 alpha phosphorylation, because of which there is a decrease in the translation. And there are multiple kinases which are known, like GCN is the one which is under the nutrient starvation condition, but there are kinases like PERK, PKR, and HRF which sense different stress conditions, and they lead to the inhibition of the translation initiation via phosphorylating EF alpha. And this is also in the field known that when you have EF alpha phosphorylated, there is a highly chance that non-canonical translation can be increased. So that's why now, because in this condition, since we also see this EF alpha phosphorylation increase, we are interested in exploring what kind of non-canonical translation undergoes. So we are still, like, since it's 1.5 years for us, still in the process of establishing things. But we will be using multiple approaches like ribosome sequencing and ribosome profiling, proteomics, and nascent translotom, etc., to investigate what all the uh, proteins which are made apart from the known mechanism, and then see whether do they have really any contribution in the disease progression. So these are the people who are now exploring this mechanism. Few more are there. So yeah, this is what the main crux of our lab that we are interested in exploring. So that so to summarize, what we are in general interest in the lab is to study the translation reprogramming, how the translation remodeled in disease condition, both in stress context as well as the mTOR regulation context, using yeast as a model system as well as HCC as a model system, and understand the and trying to discover something new pathways in this context. So this is it. Thank you all. I think that's it. This is my group, which keeps changing a bit, but some core members are there. I think, yeah. Can I stop staring?
Yeah. Yeah, sure. I think it was a really good wrap up to mTOR signaling that we studied during the masters.
Masters.
Yeah, but like since I'm in the Cancer Research Institute, so we usually see mTOR signaling and
Yeah,
dopamine.
many of the contexts you will see it. Yeah,
Yeah, true,
that's why
true.
it sometimes it's a tricky because you whatever you do, you see a effect on the end. So
Yes, that's true. That's true. Sir, so like I would like to ask like one really basic question because I'm not in the field. But how do you necessarily identify the initiation of the non-canonical translation versus the canonical one? Like, are there any markers for that?
that's what. So for that, you have to do this ribosig technique.
Mm-hmm,
So
mm-hmm,
ribosome profiling technique where we actually isolate the ribosomes
mm-hmm,
and
mm-hmm,
their protected fragments, mRNA fragments, and
mm-hmm,
sequence it. So that's the
mm-hmm,
way where we can identify the non-kinetic. Like if the ribosome is out of here, their known openating frame, you
mm-hmm,
can trap that using the ribosome
mm-hmm,
sequencing, ribosome profiling technique. So that's the thing which we are now trying to establish. But there are tools like you can use some drugs to trap ribosome only during at the start codon and
mm-hmm,
again sequence that. So, but at
mm-hmm,
that you depend on the sequencing technique to identify non-can. Once you know that, then you can go for the generating reporters or something. But
mm-hmm,
before that, I think we need this information first in
mm-hmm,
cancer. What all the non-canonical translations undergoing. So that's the idea. Yeah.
mm-hmm. I see. I think that's a really cool idea. And like I remember this polysome profiling and translation initiation because I was working in ISC for my master's thesis,
Okay.
and I was in East Lab itself.
Oh, which department?
in biochemistry, Dr. Purushat Rajaguru.
Acha. Okay, cool. Yes, the Rajguru. Okay, nice,
Yeah.
nice.
So I
Yeah,
did
so
my
they did polysome profiles. They
yes,
had their own. Yeah,
they have small machine to
yes.
make the polysome profiling, and I mean I never did it myself for polysome because that was not the project, but yeah, it was interesting, difficult as well.
Ah, success rate is 50%, I would say. My students struggle now, so I keep them that. Don't expect too much. 50% if it works, then it's you're happy. That's
Then
good
it's
enough.
good.
Yeah.
50% is also too.
True. That's true.
I think
That's what you have to
20%.
be happy.
Yeah, I think that was a really brief presentation where I'm sure students would have understand mTOR signaling and also how to target it. The double
Yeah, I
network
hope
story.
they understand. But if they have doubts, they can write to me anytime.
Mm-hmm. So, like I ask this question usually to all the scientists. I mean, two questions which we usually host them. So one of them is like, if you could achieve like a major breakthrough in your life, then what have you dreamt? Because I'm sure every scientist, whenever
Wants
they,
to find.
yeah.
So my imagination is that with these projects, okay, my aim is to identify something which is working for the only for the cancer cells. Okay, because the thing
Mm-hmm.
is, these normal cells do not should not be very much dependent on this such a alternative mechanism. So
Mm-hmm.
only under stress conditions, these alternative mechanisms should be active. So
Mm-hmm.
we are hoping that we might be able to discover something which is very specific to HCC cells, cancer cells.
Mm-hmm. Mm-hmm.
And then that's another thing that we are also trying to use the AAVs as a gene therapy model.
Mm-hmm.
So in future, if we can able to target that specific,
Mm-hmm.
that's good enough for me. I don't have to think too
Mm-hmm.
much about it. So, but yeah, getting that one target, you might make multiple stories definitely, but a one which is really going to work is, I think, a challenge.
Mm-hmm.
I think it will take time. So
Mm, I see.
let's see how it goes.
And how do you envision yourself in next 10, or I would say 10, 15 years from now?
Maybe I don't know. Maybe I will become a little bit grumpy. But yes, said that I don't know in the next 10 years because, as I said, it depends on my students. At
Hmm.
that.
Hmm.
It's not that I will drive everything in
Hmm.
my lab.
Hmm.
So my
Hmm.
students will drive. So I don't know what next comes. Like I'm excited with my students that they will have creative ideas and all.
Mhm. Mhm.
So the lab shape will depend on that part. I will say.
Hmm.
Definitely, I will have this whatever the program started. We'll have that. But
Hmm.
at the end, depends on what we will be discovering in near future.
Hmm.
Based on that, you will have. But strongly, we are trying to get this gene therapy aspects a little bit more functional.
Hmm.
So hopefully that part, and then we are also in some communication industry setup. So
Mhm. Mhm.
that might help into a little bit of the other applicative part. So
Hmm.
that one thing.
Hmm.
But otherwise, I think there are a lot of questions open because this is one part, but there are multiple other aspects of the protein synthesis which we are interested.
Mhm.
So I
Mhm.
think I see that multiple like things which I can handle, maybe not able to handle,
Mhm.
but yes,
Mhm.
yes, it will be exciting, I would say.
Mhm.
But I can't say that one thing which will happen in the next 10 years. We don't know.
Yeah. Okay. Okay. So one more question, and then I would like to take up the audience question, which are
Oh,
being
do
a
they
lot
also
of
ask?
mTOR signaling questions. So one last question from my side would be
Okay.
that uh, what is your favorite discovery in science so far?
So far,
Yes,
so
like.
far, okay. Like for me, myself, like the second paper, like the third paper, what do you say? In my PhD, is my favorite discovery in the sense where we were showing that like ribosome biosynthesis happens, right?
Mhm.
So, and ribosome biosynthesis is a very complicated process. Like you have so
Mhm.
many factors coming, binding,
Mhm.
all the secondary structure has to
Mhm.
be done
Mhm.
properly and all. So then question is how does the cell knows what is such assembly, whatever happened,
Mhm.
it's really properly done or not? You know,
Mhm.
that's the question still open. Right? It's just a
Yeah.
macromolecule has to functionally assemble and then you have to
Yeah.
ensure that okay, it is proper. So
Yeah.
one of my study where it was like a serendipitous observation, like I don't know, but it was experiment made
Mhm.
for something else. But then we started with that observation that uh, the native, the ribosome, the
Mhm.
initial, the once it's fully formed,
Mhm.
okay, and that ribosome has to do a job of initiation,
Mhm.
undergo first round of initiation cycle to be said that okay, now I am fine, I am doing my job properly, so now
Mhm.
I can go further next, like I can repeatedly do that job. So
Yeah.
that quality control is at the initial step is what we propose. That was a
Mhm.
PNA's paper. So and we propose that mechanism that the whatever the newly synthesized ribosome has to undergo a first round of initiation to be qualified that okay, you are fine now, you can be given to the proper functional pool of the ribosome. So that hypothesis we threw in that study, and that is still my favorite because my boss was not agreeing that concept for 6 months initially. Like he was like, okay, this data doesn't look like other. So I had to argue with him, and I had to get more and more data to support that aspect that indeed it is the mechanism happening. Otherwise, you will accumulate these immature ribosomes which are
Mhm.
not properly folded. So that happens like that. But then I think that is well accepted now a little bit. So I think it's nice. So that study I really still
I like
liking it.
it.
But I think best one has to come still.
I think I would look after that paper.
Hopefully, yes.
Okay, okay. So there are few questions, so I would like to pick few of them
Yeah,
from
that's
Gaurav.
better.
So one thing that is an interesting question to me as well, that how can proteomic profiling help predict patient response to mTOR inhibitors?
Yeah, that's a big challenge also, you know.
Mhm.
For that, we don't know the answer, okay? But we can see that if you do the phosphoproteomics, and
Mhm.
if we
Mhm.
find it's not that all the patients will have a mTOR hyperactivation,
Mhm.
certain
Mhm.
population. And
Mhm.
if we see with the phosphoproteomics that signatures of the phosphorylations are high for the mTOR related pathway,
Mhm.
then we can think them as a most likely a responders
Mhm. Mhm.
because those cancer cells might be addicted for the mTOR pathway. So we can think about that as a good response. But this kind of study is has been not done so far. Okay,
Mm-hmm.
so that's one aspect we can think about. We do that and then do the treatment, but then see how they respond and how, because already some of the MAPK inhibitors are there. In the liver cancer, at least, MAPK inhibitors are treated as a last, like if the surgery doesn't work, radiation doesn't work, then they use as inhibitor. It's a very toxic. But then again, they also develop resistance within 2-3 months. Some because growth factor is very compensatory, like too many intertwined there, so difficult to. So mTOR also, I feel that it will be compensated by other pathways, but we need that study. So, but yeah, phosphoproteomics we can do and see which patients have this hyperactivity. That will be the answer.
I see. I see. I think that's pretty convincing. So, also, like, what are the current challenges in like proteomics contributing to mTOR targeted therapies?
So the proteomics, the current challenges are the main challenges, the tumor heterogeneity. That's the main thing, because if we are going for the bulk proteomics, we don't know what's happening where. Okay, we assume that okay this, but mTOR might be hyperactive in the stromal cells, not in the main cancer cells. So multiple things are possible. So nowadays people are more into spatial proteomics. So that's the one advancement is going to happen in the near future. Spatial proteins will be more. Right now it's majorly for the metabolites and all, more reliable. For the proteomics level, it's not so reliably standard, but it might happen too soon. But then that might give us a better idea about both overall environment of the tumors and the the where to target, which population actually is the responder. Just doing the bulk proteomics may not help us.
Yeah, yeah. I think makes sense. Good. So we have other questions as well. One really basic question: that if puromycin is competing with tRNAs, where it is incorporated in the peptides?
At the end of the peptide, like it's competing with the A side, since it's
Yeah.
binding to the A side,
Hmm.
so it will be at the end of the
Yeah.
any amino acid. It doesn't have, because since it doesn't recognize any codon. Once you add within 5 minutes, you incorporate most of the puromycin in all the peptides. So as such, no. But people have said that it's somewhat mimicking to the tyrosine codon, like amino acid. But it doesn't matter, because at the end it doesn't recognize any codon. You just go to the A site whenever it's empty and ligate it. So randomly everywhere in the protein, you get that puromycin incorporation.
I see. Okay, so I would just quickly ask one last question for today,
Yes.
and it is: how do you relate spatial proteomics with cellular level of metabolism?
Cellular level of metabolism. That's what. If that's a, like, I don't know whether the person wants to do a both spatial metabolomics and the spatial proteomics.
Yeah.
Definitely, that will be a cool idea to go for it. Like, since we have the sections, we can go for the metabolomics as well as the proteomics. But definitely, both are important and both can give a more insight. Yeah, we also have, like, I don't have expertise right now for those parts. But definitely, we are right now thinking of going for the individual metabolites to screen, which we are interested in. But we can think about the global level. So that's what mainly the issue with right now is those, like the reads, how much you get, like the complex samples, difficult to segregate with each sample, or like each spot. If you go for the target, it's very easier, it's feasible. But if the complex ones, for the proteins and metabolites, bit difficult, but it's possible, I would say, and it will have more improvements in near future.
Good. Then it was, I would say, a really nice talk
Thank
today.
you.
Thank you so much, Dr. Sunil, for coming today, taking time out, explaining us mTOR signaling on Sunday morning.
A little bit, not so much depth, but fine. But thanks a lot, Saicom Siddhas, for organizing this. I think many students get benefit with this.
Sure.
And I would say that all the students that if you are really genuinely interested in research,
Hmm.
have patience. Okay, three things I wanted to give a message for the students that one is patience and don't lose the hope. Okay,
Hmm.
in the research, you will have a negative results lot of time, but
Hmm.
you have to keep your motivation up. That's why I said always do a multiple projects so that your curiosity will remain like the awake. That's
Hmm.
one thing.
Hmm.
Second is networking is important in science. So
Hmm.
keep expanding your networking, interact with
Hmm.
people, network it. That's more important for all the students
Hmm.
because that helps a lot in your career,
Hmm.
networking. And then ask more questions. Nowadays, students don't ask so much questions during any meetings or anything. So I would say start asking questions, then your brain gets more and more involved and you will enjoy it. So these three qualities they have to develop. Most of the students should do that. You will enjoy the science field, definitely. And good luck to all the students who are attending. That's good. Thank you, Dairya.
Thank you
Thanks for
so
hosting.
much, Dr. Sunil. And I would also like to thank all the audience today. We would send the feedback form link. So, Dr. Sunil, we usually have the feedback form link for the talk, and
That's good.
people
I
can
like
suggest
to know.
us. People can suggest us how we can improve more, because we also want to take it ahead, the initiative, and host more and more PIs, and let students get introduced with the young researchers in India, right?
Yes, that will be great. Okay. Thank you very much. Good luck to all. Okay.
Okay, we would send the feedback form link uh, right now. Please fill it for the e-certificates. E-certificates would be sent to you via email only if you fill the feedback form. So please fill the feedback form right now.
Can you pin me, please? Darya, pin me. Make it full screen, my screen. So, to all the SciComm Synapse audience, we have a very good news today for you all that we have completed 500 subscribers on our channel. So, it would be really great if we can get your support, your your motivation, your inputs, so that we can improve a lot, and then we can reach to the success, the summit of the success, and we can provide many more information and insights to our audiences as well. So till then, please do like, share, and subscribe our channel so that we can soar the sky. Thank you all and have a good Sunday. Bye bye everyone. Do like, share, and subscribe.
Okay, we would give you the accesses for the feedback form, and you would receive the feedback form in your Gmail account. So please fill in the details in your Gmail. So you can request the access, and then you will be given the access for this, and you can fill in the feedback form. Yes, everybody would be given the access to the feedback form. Please, you will get it in your email, Everybody will receive the access. Okay, thank you so much.
Yeah, can you definitely check once? Are we are the audience able to open the feedback form? Can you please check the same link?
Yes,
Can you please check? Tell the audience to that post a yes if they are able to.
yes, now they can fill it. So, yeah, I think it's open. Okay, so thank you so much. We have provided access to everybody.
Yeah,
We
audience,
have,
please check on the same link that are you able to fill up the form? Audience, please do check once, then so that we can end the live. Still not?
I could open the access completely. I could completely access the form. I mean, yeah, everybody could access the form. Please try to uh, click on the link again. Okay, so we would end the live stream here. Please fill in the feedback form. The link is shared in your Gmail. Bye bye.